We're almost at early semester, mid-semester break. So today we're going to finish up the diffusion and transport of carbon dioxide by blood, and then get to how the respiratory system is controlled by the nervous system. So this was the slide that was on the board at the end of the day on Thursday. Oh, by the way, problem sets will be ready maybe by the end of the day today, definitely by the end of the day tomorrow. I don't have them yet. So they'll be done graded and you can pick them up by the end of the day tomorrow for sure. And then plus you may be aware that I'm posting grades on Moodle, so if you're interested in your grades, you can go to Moodle. Remember that there is no lab on Thursday, of course, it's break, and there's no lab next Thursday because that's the exam day. So exam 2 is a week from Thursday, and the same schedule will apply next week as applied for the first exam. In other words, you can take it during class time or you can take it during lab time. Okay, so let's not just pick up where, let's talk about this slide. So carbon dioxide is produced by the mitochondria of metabolically active tissues, right? I just taught this stuff in Self-Phys, so it's on my mind. What are the steps that produce CO2 in cellular respiration? Well, oxidation of pyruvate produces CO2. The Krebs cycle produces CO2. Those are the things we talk about in Self-Phys. Oxidation of fat and protein also produces CO2, although the steps are slightly different than the oxidation of glucose. So, and that's in the mitochondria. Mitochondria of metabolically active tissue produces CO2. CO2 diffuses from the mitochondria into the cytoplasm. CO2 diffuses from the cytoplasm through the cell membrane. CO2 diffuses through the cell membrane and is dissolved in the extracellular fluid or interstitial fluid around whatever tissues we're talking about. So we got steps of diffusion. Diffuses through the endothelium of the capillary that is perfusing whatever tissue we're talking about. So we're now in the capillary. Diffuses into the blood plasma, where some of the CO2 is dissolved. I mean, it's dissolved everywhere, but some of this CO2 that diffused there stays there as dissolved CO2. Some of it diffuses, so now I'll start labeling because I think this is where we left off. Some of it, this is a step that is diffusion. It's another diffusion step. Diffusion into the cytoplasm of the red blood cell. So through the red blood cell plasma membrane into the cytoplasm of the red blood cell, where some of it stays dissolved. But some of that dissolved CO2 binds to hemoglobin. And that binding, remember that binding of CO2 to hemoglobin is one of the things that helped the hemoglobin let go of its oxygen in this metabolically active tissue. But we're focusing on CO2, so we're not seeing the oxygen right now. So some of the CO2 binds to hemoglobin. It doesn't bind at the same place where it's not a competitive inhibitor of oxygen binding, but it facilitates oxygen unbinding from hemoglobin. Some of the dissolved carbon dioxide combines with the water that's in the cytoplasm and makes carbonic acid. Now this happens out here too, But this step is accelerated about 1,000,000-fold by the presence of carbonic anhydrase. Carbonic anhydrase. So you probably can't read that from the back of the room, but you should annotate your handout with what Ca stands for, carbonic anhydrase. It's an enzyme. It's present inside red blood cells. And as an enzyme, it accelerates the reaction. And in this case, the reaction that it accelerates is the combination of CO2 with water to make carbonic acid. Accelerated 1,000,000 times by carbonic anhydrase. So out here, that step is very slow, and here that step is very fast. Carbonic acid dissociates, so this is just dissociation of an acid into protons and bicarbonate. Bicarbonate, so the strong acid, the weak base, bicarbonate base. Okay, so we have converted some, maybe most actually, of the CO2 that gets inside the red blood cell is converted into bicarbonate ion. Some of it is bound to hemoglobin, and some of it stays as dissolved CO2, but a lot of it got converted to bicarbonate ion. Now, here's a step that your book is particularly sloppy about, because this step, this arrow looks like bicarbonate is just moving out of the red blood cell. And this arrow makes it look like chloride ion is just moving in. Well, neither of those ions just diffuses, right? Because they're charged. And you know that ions don't diffuse through plasma membranes. Your book is really sloppy about this because there is a transporter, an exchanger, that has the very helpful name, the chloride bicarbonate exchanger. Tells you exactly what it does. The chloride bicarbonate exchanger is a protein. It's an antiport protein in the membrane of the red blood cell that allows bicarbonate to flow out down its concentration gradient in exchange for chloride. So it's neutral. But the point is, some of this bicarbonate that got made in the red blood cell because it was accelerated 1,000,000-fold by carbonic anhydrase, now is being transported by the blood plasma. So the blood plasma and the red blood cell cytosol are both carrying carbon dioxide, both in dissolved CO2 form and after conversion to bicarbonate ion. So the carbon dioxide that's carried by blood is carried by red blood cells in the form of CO2, bound CO2, and bicarbonate ion. And it's carried by the blood plasma as dissolved CO2 and bicarbonate ion. Some of you, does anyone know what a typical hematocrit is? You know, what percentage of the red blood cell, what percentage of the blood volume is red blood cell? It's about 38%. So, and I'm not going to hold you to knowing that number, but the point is 2/3 or so of your blood volume is not in red blood cell, it's out here in plasma. And so this carrying of bicarbonate by plasma and this carrying of dissolved CO2 by plasma is an important part of the carrying capacity of blood, the capacity of blood to carry CO2. Okay? So we're carrying CO2 in multiple forms, and this slide tells you the story of how we get that CO2 carried in blood. Any questions? Okay, so now blood flows to the lungs. So all of this all of the process that I'm going to tell you about here is really the reverse of the process that we've gone through. So I'm going to go through it slowly, but it's all familiar to you now. And it's all being driven by the fact that the carbon dioxide partial pressure in the alveoli is low relative to the partial pressure of CO2 in the venous blood that is perfusing the alveoli. And the CO2 is low because we ventilate it out. So this is ventilation. So let's go backwards. Let's work our way backwards. Ventilation of CO2. Alveolar ventilation. Things that you worked with on the problem set. What is this arrow going? What is this arrow? Diffusion. What are some of the shown and not shown cells that this is diffusing through? Well, okay, what is the name of the cell type that lines this capillary in the blood, in the lung? Endothelial cell. So this is an endothelial cell. What's the name of the cell that is present here that is just not shown in this oversimplified cartoon? What are these cells here that line the alveoli? Type 1 alveolar cell. Okay, so we've got diffusion through thin, specially thin cells. Good, okay. So this arrow is diffusion. It's diffusing because some of the CO2 is dissolved in blood plasma. Some of it was diffused from blood plasma from the start, but now let's work our way back up some of these arrows. Some of it, so what is this arrow that is this arrow here that is taking dissolved CO2 to dissolve CO2 here? What's this arrow? Diffusion. Some of this CO2 was not from, well, where did this dissolved CO2 come from? Well, some of it was dissolved from the start. But some of it was not. Some of it came from hemoglobin. So what would this arrow be trying to describe? Diffusion. Okay, so diffusion, but there's one other step. There's another step. How do we get from this to this? Diffusion is part of it. Dissociation or unbinding, yeah. Because here the carbon dioxide was bound to hemoglobin, and here it's free dissolved, and so there had to be an unbinding step included in this arrow. Some of the CO2 came from bicarbonate. So let's watch this. So so again, your textbook is defective or deficient. What is this arrow? What is this blob? It's the exchanger, the chloride bicarbonate exchanger, running in the reverse direction from the way we had it over here. All exchangers can run in both directions. And so now we have it running where bicarbonate is moving down its concentration gradient from cytoplasm, from plasma to cytoplasm. We have a binding step. So this is, if this is dissociation, then this is association. Association of bicarbonate with protons, forming carbonic acid, which can be broken down. Now we're talking about breaking down by carbonic anhydrase into water and CO2. We're just reversing all of the enzymatic and transport steps that we understood over here. So all of these, and this is all happening because the CO2 is flowing down its concentration gradient by diffusion. So if this CO2 is moving out here, then this CO2 is moving down here, then this bicarbonate is moving here, here, and here, and the whole process is going down its chemical equilibrium, down its chemical concentration gradients, to produce CO2 that we ventilate out. To produce, we're producing the CO2 either from dissolved CO2 or from dissolved bicarbonate. So we got a mess here on the board, but I think I hope it's clear the steps of the story of carbon dioxide transport by blood, or production by tissues, transport by blood, transport and by lung capillaries, and then ventilation by lung alveoli. That was the goal of this story. Are there any questions? It's got a few more steps than oxygen. Oxygen is simple. It basically breaks down to knowing some anatomy, knowing the names of the cells, and knowing that oxygen diffuses and binds to hemoglobin. Oxygen is simple. Carbon dioxide has more steps because we have both the dissolved CO2. We have dissolved oxygen too, but 90-something% of the oxygen is carried as a bound form. Here in CO2, it's less dramatic, but much of the CO2 is carried as the bicarbonate ion, and we have to understand where that bicarbonate ion came from. It came from carbonic anhydrase and and its presence in the plasma came from the chloride bicarbonate exchanger. Questions about this? Okay, so it's just a matter of, my recommendation for learning this is to take the blank slide and, annotate it again to tell the story. Okay, so let's move on to the control of respiration. So here is your friend, the Davenport diagram. So we're talking about respiratory acidosis, say during hypoventilation, or respiratory alkalosis during hyperventilation, or we're talking about respiratory compensation to metabolic acidosis, or respiratory compensation to metabolic alkalosis. So we're talking about the control of the respiratory system. So so what is controlled? Well, the things that are controlled are blood pH. So the variables that are controlled are blood pH, blood partial pressure, arterial blood partial pressure of O2, and blood arterial partial pressure of CO2. And these things are highly related because of the equilibrium that you know. Carbon dioxide goes up. We drive this equilibrium to the left, to the right, and pH goes down. So those are the things that are detected. How does your body detect it? Well, we have chemosensors, neurons that detect chemicals. The chemicals being acid, protons, oxygen tension or oxygen partial pressure, and CO2 partial pressure. They're in the same place. At least some of them are in the same places as the baroreceptors that you might remember from the circulatory system that control that detect blood pressure and control cardiac output and control vascular resistance in order to control blood pressure. So the anatomical locations are in what are called aortic bodies, cells that are in the aortic arch, and cells in the carotid bifurcation, the split between the internal and the external carotid arteries. Remember this, so let's do a little anatomy. There's the aorta, that one's labeled, aortic arch. What's ventricle? Left ventricle is what pumps blood out the aorta. What's this? Superior vena cava, inferior vena cava. What's this? Diaphragm, right? We've still got to remember we're now in respiratory, but we don't even see the lungs in this picture. What's the name of this vessel? Brachiocephalic, which slits into the common carotid and the right subclavian. Good. And here is the left common carotid. And here is the left subclavian. So the two common carotid arteries, both the right and the left, split into the internal and the external. And at that split is where carotid bodies are that are neurons. They detect chemicals. And that's the yellow lines here are the nerves that take action potentials from those sensory cells to the brain. So this story here that I've been telling are the peripheral chemosensors. These are the peripheral chemosensors in the aortic arch and in the carotid bifurcation. So this slide also indicates that we have central chemosensors. The central chemosensors, which also detect chemicals, chemosensors, are in the hypothalamus. And since this is not, so those of you who have taken A&P1 have learned a lot of the neuroanatomy here, and you can apply it if you want, but I'm not going to teach neuroanatomy in A&P2, just to know that but you do need to know that the hypothalamus, a part of the brain right here, is the location of the central chemosensors. That's the key point. So let's walk through, here's a flow chart. Let's walk through this flow chart which is the control mechanism for respiration. So let's start with a focus on detecting pH. Because pH is going to be our proxy for detecting CO2. Because if we detect protons, we're indirectly detecting CO2. So here is the mechanism for both central and peripheral detection of pH/CO2. Okay, so let's just walk through this. Alveolar, if alveolar PCO2 increases, what are we talking about? Are we talking about, well, what are we talking about? If alveolar PCO2 goes above normal, what are we talking about? Hypoventilation, which is associated with respiratory hypoventilation, high PCO2, hypoventilation, because we're not exhaling enough CO2, hypoventilation is associated with respiratory acidosis. So alveolar PCO2 goes up. As a result, arterial PCO2 goes up because they're almost always equal. Okay, so now let's follow the peripheral line here. This is the peripheral out here. So if arterial PCO2 goes up, then arterial proton concentration goes up because of the equilibrium that you know about. And so arterial pH goes down. That's respiratory acidosis. And there are sensors in the peripheral chemosensors, in the aortic arch and the carotid bodies, that detect this acid and increase the frequency of firing of action potentials. We'll get to the mechanism of that in just a few seconds. So we have an increased action potential frequency going to the medulla. So again, I'm not going to teach much in the way of neuroanatomy, but those of you who've had neuroanatomy should know that the medulla oblongata is the place where this control is done. Increased firing of input, increased firing of the output neurons that innervate the diaphragm and the external intercostals, leading to an increased ventilation, trying to return PCO2 to normal, which returns pH to normal. This is respiratory, compensation for respiratory acidosis, homeostasis. The same story is true in the central in the central chemosensors. The same story is true. PCO2 goes up, pH goes down. Everybody clear? When this arrow up, brain extracellular fluid proton concentration goes up, so pH goes down. Got to be facile with that, with keeping track of that. So again, we have firing of chemoreceptors in the hypothalamus that then tell neurons in the medulla oblongata to fire. Excitatory synaptic transmission for those of you who are interested in neurons. More action potentials in the nerves that innervate the diaphragm and the external intercostals, and more ventilation. So what are the key points here? The key points are that the central and the peripheral chemosensors detect acid. And that detection then is integrated in the hypothalamus and sends input to the medulla to control respiration. And so in the case of, well, let's just walk through. What if alveolar PCO2 goes down? What do we call that? Let's just do the opposite. That's not illustrated here, but we have a decrease in alveolar PCO2. Hyperventilation or respiratory alkalosis. So respiratory alkalosis means the pH is high, so the proton concentration is low, and that would lead to a decrease in firing. which would lead to a decrease in the contractile output, which would lead to a decrease in ventilation. That's the main point here, is that pH is detected and that the hypothalamus and the medulla are the control centers that do the neuronal regulation. There is a nerve, so the main nerve that goes from the medulla to the respiratory muscles is called the phrenic nerve. The phrenic nerve innervates the diaphragm. More action potentials in the phrenic nerve, stronger, more frequent contractions of the diaphragm. I'm not going to hold you responsible for that neuroanatomy. It just is. So here's a clicker. Let's activate a little bit here, do a clicker. So this is just a piece of, this image here is just a piece of the previous slide. Peripheral and central chemosensors, peripheral and central chemoreceptors, contain a channel that detects pH. It's called ASIC, for acid sensing ion channel. It's a channel that senses acid. So we're getting now into the molecular mechanisms of how a neuron can detect acid. It's got a channel that detects acid. A channel that detects acid and causes the neurons to increase their action potential rate. Now, I know I didn't teach neurons, but I did teach threshold, and I did teach action potentials in the heart, and the principles are the same. So now I would like you to figure out what ions go through acid-sensing ion channels. Acid is the thing that they detect, but acid is not the thing that goes through them. So most of you all of you, I think, all of the ones, I'm seeing mostly correct answers. The correct answer, the simple correct answer is A. You could have said, well, they could be permeable to both sodium and potassium, because that would also cause the neuron to depolarize. That's a true statement. It turns out that they are selective for sodium. If you chose potassium only, you were wrong, because if you activated a potassium channel by acid, that would cause the neuron to hyperpolarize, and that would decrease the firing rate, not increase the firing rate. Right? That's the concept that I'm trying to get you to know here. Acid-sensing ion channels cause an increase in the firing rate. That means they bring the cell to threshold more, which means they depolarize more. And the thing that causes depolarization, we're going all the way back to the beginning of the semester, is influx of sodium. So these channels are sodium selective. So the peripheral and the central chemoreceptors express a channel that opens in response to acid and causes depolarization, which causes increased firing. And now we get into some complicated reflex neurophysiology, but it doesn't have to be complex. Increased firing of these input neurons and an excitatory synapse just means that we have increased firing of these output neurons and increased ventilation. And the flip is true if the arterial pH goes up, pH goes up, so proton concentration goes down, less activation of ASIC, less sodium influx, less depolarization, less firing, less contraction. Okay, so that is the main story of control of respiration by the brain. Are there questions about that? There's another partial story that I'm going to introduce in the next slide. But this, if you get this, you're 90% of the way there. So we also detect PO2. So the point is we detect PCO2, but mostly by detecting the acid that the CO2 produces. Acid becomes the thing we detect in order to detect CO2. We also have oxygen sensors, but they are located only in the peripheral chemosensors. So see what's happening here. The central chemosensors and the peripheral chemosensors detect acid. The central chemosensors detect only acid, and the peripheral chemosensors detect both acid and O2. So it turns out that the peripheral chemosensors do have mechanisms for detecting oxygen, and it's actually a complicated metabolic thing. More oxygen, more metabolism. More metabolism means the neurons change their behavior and they fire more. In fact, I'm rusty on the molecular mechanisms that lead to this, that this arrow leads to this arrow. But it's true, and it has to do with metabolism. So this arrow is asic. This arrow is asic. And this arrow is still kind of a black box, but it's there. Is it clear what I'm trying to get at? We understand the molecular mechanisms for this and for this. And although we, as a field, we understand the molecular mechanisms of this, I'm not teaching it to you because it's complicated. And this figure, which again from your text, specifies that ventilation is due to inspiratory neurons that increase the contraction of the diaphragm, and this inspiratory intercostals are the external intercostal muscles. So all of these contractions are increased, but this is all embedded. Well, no, this is embedded in increased ventilation. What's the main point? The main point is both peripheral and central detect CO2. Well, really they detect acid, but the acid is a proxy for CO2. And peripheral also detect oxygen, but central don't. Question?
When do we have the lower partial pressure of oxygen? Is it the case when it starts to compensate the increase of partial pressure of CO2?
Well, so it would a decrease in arterial PO2. Is that hyperventilation or hypoventilation?
I think in the previous clicker question, when not in this class, like exercise, I think it was about exercising.
Ah, okay. So if we, well, let's go through this arrow. So if there is a decrease in arterial PO2, is that hyper or hypoventilation? Where was I? Oh, a decrease in arterial PO2 would be indicative of hypoventilation. So now tell me your question again.
I was wondering because in the Davenport diagram we don't see PO2.
So the Davenport diagram doesn't show us anything about PO2. The Davenport diagram is only about pH and P CO2 and bicarbonate. So the Davenport diagram, you can't use the Davenport diagram to get any information about PO2, except that if there is hypoventilation, there is also going to be a decrease in arterial PO2. If there is hyperventilation, there may or may not be. I mean, if there's hyperventilation, there should be a small increase in arterial PO2. But you should understand why it would be small. Anybody want to venture an answer to that right now? Why would PO2 go down substantially during hypoventilation, but PO2 would go up only slightly during hyperventilation?
Drawing in as much oxygen.
Say that again?
You're not drawing in as much oxygen.
Well, no, that's not the answer. Me?
You're ventilating so fast that the exchange of gas in your capillary has not become.
No, that's not really the answer either. Anybody else? I mean, those aren't wrong, but those aren't the answer to my question. Hypoventilation causes a significant decrease in PO2, but hyperventilation causes a very, very slight increase in PO2. Why?
Because like oxygen is like mechanism to compensate.
No, it's not that either.
Is
Jason?
it because the blood can only become so saturated with
Where's the
oxygen?
answer? The hemoglobin, even under normal ventilation, is 97% saturated. So if you hyperventilate, you can only go from 97 to 99% saturation, and PO2 stays about the same. Whereas you hypoventilate, now you're working your way down the saturation curve, and you can have a substantial decrease in PO2. So the image that's in my mind, that should become in your mind to answer that question, is the image of hemoglobin saturation and the fact that arterial PO2, so this is normal PO2, and it's 97% of its maximum. So if we increase ventilation, we can only increase PO2 by a little bit. I'm sorry, this is not. Well, this is, yes, this is saturation, and this is PO2. We can only go from here to here. And so we can only increase PO2 a little bit. But when we hypoventilate, we can go all the way down here, and we can decrease PO2 by quite a lot. It has to do with the fact that oxygen is carried mostly by hemoglobin, and hemoglobin is mostly saturated under normal breathing. Is that clear? Okay, so let's finish this up, and then we'll be done early for the day, and A and P will start your mid-semester break. So this is really for interest only. This is not the kind of information that would be on the test. Everything I've told you so far today could be on the final exam, but not this, okay? So this is a cartoon of the medulla oblongata. It's just a cartoon of the medulla oblongata. And it's using some of the neuroanatomy that some of you have learned. The point is that there are clusters of neurons in the medulla that drive inspiration, and there are clusters of neurons that drive expiration. So let's just, let's not try to make too much sense of this. But just notice that there are neurons that cause the diaphragm to contract. So this is the phrenic nerve, a nerve that I introduced before, and that innervates the diaphragm, and that causes inspiration. The diaphragm contracts, it flattens, and that expands the chest cavity, and that decreases intrapleural pressure. I'm doing your problem set for you, and that decreases alveolar pressure, which draws air into the alveoli. So that's inspiration. And here's some more nerves that drive the external intercostals. So these are nerves that drive the muscles of inspiration. But notice that there are other nerves. So what do you suppose cranial nerve 11? It's called the accessory nerve. What are these other muscles? Sternocleidomastoids, scalenes. These are the muscles that are, this center is recruited in the case of needing to ventilate, to inspire more. And you have centers in your medulla that cause you to start mouth breathing. You know that if you need to ventilate a lot, you don't keep your mouth closed anymore. You now start to breathe through your mouth. And you may not even be aware of that you're opening your mouth to breathe because it's controlled by the medulla oblongata, which is an autonomic part of the brain, automatic. So you automatically open your mouth and start mouth breathing in addition to nose breathing. And in fact, you may not even be aware of it, but you use muscles to try to expand your nasal cavity, to flare the nostrils. So this is all controlled by the medulla oblongata. It makes sense. Some of it you know, increasing diaphragm contractions, increasing external intercostals, increasing sternocleidomastoid contraction if necessary, opening the mouth for mouth breathing. It's all done by the medulla oblongata. The expiratory centers in the medulla oblongata tell the abdominal muscles and the internal intercostal muscles to contract if necessary for forced expiration, but only if necessary, right? So these centers are recruited only in the case of, needing to exhale forcefully. It's actually a really interesting nervous system because it oscillates. There's the inspiratory neurons fire, and those inhibit the expiratory neurons because you don't want to do both at the same time. And then the and then these turn off and these turn on. So there's a handoff between inhalation and inspiration and expiration. And then there's all these other effects of breathing. And a lot of neuroscience is actually dedicated to understanding the details of respiratory neuroscience, the control of respiration by the brain. I think that's it for today. So, enjoy your break. Think about your breathing. As you're breathing, maybe be aware, okay, what am I doing? What's going on? If you want to pick up your problem sets, I'll have them by the end of the day.